let me turn it over next to Dr. Koch. Thank you.
Thanks very much. It's a pleasure to be in Washington. I spent quite a few years at 200 C Street with the FDA, just a couple of blocks from here, brought back some fond memories. I left in 1998 to join the molecular diagnostics industry. Roche Molecular Systems was formed when the PCR patents, polymerase chain reaction, were acquired from CETUS, and a whole new industry was started in molecular diagnostics. And I wanted to be part of that revolution, particularly this notion of personalized healthcare. The story I'll tell you about today is exactly that, marrying a diagnostic result to a therapeutic selection that has tremendous benefit for patients. So I won't spend a lot of time on this. I think you understand the general concept. Dr. Issa already told us that we've been moving towards a molecular-based definition of disease. And that's where diagnostics come in, to tell us that a tumor that sits in your lung is one of perhaps 20 different subtypes that has to be treated differently. And only with molecular diagnostics can you ascertain what type of disease you have and how to best treat it. Molecular diagnostics are leveraging some 30 years of scientific inquiry into a variety of diseases, but here we're talking about cancer. And sequencing of cancer genomes over the past decade is providing us a lot of granularity about these very diverse cancer types that are defined by molecular defects in the genome that are acquired over time. So if we can use a prism, such as a molecular test, then we're going to go from a situation that you heard described earlier of a generic chemotherapy, radiation therapy approach to one where, based on your molecular profile, you get a therapy that's going to be more efficacious for you with a better side effect profile and response. And that's precisely what we're doing. It's pretty obvious that leads to more effective drugs. It leads to fewer failures in the clinic and also shorter development cycles to get new medicines to patients quicker. Who benefits? Everyone. Patients, payers, physicians, and drug developers. You can imagine, from the patient perspective, any better therapy when you've been told you have stage 4 cancer is going to be welcome news. The physicians clearly want to provide the best medicines they can for their patients, improve the efficacy, and reduce the adverse side effects. Clearly, payers want more value in terms of cost-effective health care. We as an industry want clinically differentiated medicines and tests. And last but not at all least, regulators such as the FDA and policymakers such as you in the room want to have improved healthcare cost and delivery of medicines. So it's a no-brainer. Why aren't we doing more of this? Well, there are a few challenges, and I'll share with you what some of them are and how we've tackled them. First, you have to find the right markers, so the science has to be really solid. You need to have early and significant investment in diagnostic development, and that's something certainly from the pharmaceutical development side that's been relatively foreign. This hasn't been part of their formula of drug development in the past. It changes how you do certain trial designs. Regulatory requirements vary widely. In the US, the FDA calls these high-risk PMA, pre-market approval type devices. In Europe, they're the lowest risk. You can self-certify such a test. The coordination has to occur between the two companies. And last, and also not least, there needs to be a clear incentive for diagnostic companies because, as we heard earlier, a laboratory-developed test can be introduced relatively easily without having to spend the millions that are involved in an FDA approval process and to clinically validate and prove the utility of that diagnostic in consort with the test, with the drug. So the story here is about melanoma. I don't know your experiences. I've met many people who have had melanoma. It's unfortunately increasing in frequency because we spend more time in the sun. UV light is what causes this. It damages the DNA. There are between 70 and 100,000 new cases in the US every year, so about 1 in 50 people will get it. The good news is it's on your skin. You can see it. If you see it early, you can cut it off and you're going to do fine. But sadly, a significant number of patients, perhaps about 8,000 a year in the US, are diagnosed with metastatic disease. That means it's spread. And when that happens, it's a pretty dire diagnosis. In fact, until recently, about a year or so ago, all you had was dacarbazine, a cytotoxic chemotherapy that provided no survival benefit, but it was all we had. There have been some biologics introduced, but they have very low response rates. questionable survival benefit in some cases. They're difficult to manage from a toxicity profile. So clearly, there's a high unmet clinical need here for this disease. As a result of the sequencing of the human genome around the turn of the century, it's been 12 years now, scientists started to focus their attention on diseases to ask, are there specific variations in disease tissue, such as cancer, that is causing this disease? And what they found in 2002 at the Sanger Center is that there's a kinase, that's an enzyme that puts phosphate groups on other proteins, called BRAF, that is frequently mutated in a wide variety of cancers: melanoma, colon cancer, thyroid, and ovarian. And that had some very quick ramifications for drug development as well as for diagnostics. Here is a much more stylized and very simplified signaling pathway. You saw one earlier. Receptor tyrosine kinases sit on the surface of the cell. They receive signals from other cells, and they send it down this transduction pathway with a series of switches. Each one of those proteins, like RAS, RAF, and MEK, are essentially protein switches that tell the cell grow, replicate, or stop. What happens with BRAF is it gets mutated in the 600th amino acid from one amino acid to another, and that turns the protein on so it's constitutively signaling. The switch is permanently on. That's bad for cells because now they're proliferating continuously, and that leads to cancer. These are called driver mutations. There are hundreds of different driver mutations in the various kinds of cancer, but the good news is because it's a kinase, we know how to target those kinds of proteins. Our colleagues, Gideon Bullock and others at Plexicon, a startup biotech in the Bay Area, developed a molecule based on the structure of this mutated protein that is highly selective for the mutated BRAF protein. And so they knew that this would be important for a disease like melanoma, where 50% of patients have that mutation. And they approached us in 2005 and said, can you develop a test that identifies the presence of that mutation in cancer tissue? And of course, we said yes. A year later, as we had started that development, they filed an IND. Roche Pharmaceuticals became their partner to develop the drug. And early in 2009, the test was used to screen the first patients after dose escalation. For those of you not familiar with drug development, in phase 1, when you first introduce the drug into humans, you gently escalate the dose to a point where you, before you hit toxicity, and that's where you then start to look for efficacy. What was done here was after that dose escalation had occurred, about 35 melanoma patients were tested, and those that had the mutation, as identified by our test, were treated with the drug. And let me show you what happened. About 3 years ago at the ESMO ECHO meeting, these pictures were shown. These are FDG PET images, and you can see that before treatment, there's a huge mass in the cavity, center cavity of the patient, and that after only 2 weeks of treatment, that cancer had essentially melted away. These are not typical results with cancer therapies, and so you can imagine that the community, cancer community, was really excited about this possibility that 70% or so patients might respond to this. And here, just so you have another visual, a patient who put this up on his website, all the masses that are deposited under the skin, and after 4 months of treatment, they essentially were gone. So I think it gives you a sense of the power of directing therapies to the drivers of disease and why you want to develop medicines in this way. So these results were really excited, and everything kicked into high gear. Our pharma partners quickly initiated phase 2 and phase 3 trials, and as you can see, they're only about a quarter apart. That is atypical for drug development. Normally you do a phase 2, and after you've gotten those results, you invest in the much bigger phase 3 pivotal trial that's randomized. These were run concurrently because they were so confident that the right patients were going to get this medicine and that it was going to show benefit. The trials were run globally, over 2,000 patients screened within 1 year, and the outcome was that at the beginning of 2011, the results were unveiled for the BRIM-3 pivotal trial, and the data were filed with the FDA. They were filed end of April, and on August 17th of last year, the drug and the diagnostic were approved together. It is unheard of to see, first of all, a drug have an NDA filed and then approved within 5 years, and similarly to have a diagnostic PMA approval in less than 4 months. But clearly, the FDA was highly motivated to get this medicine to patients who were going to benefit from it. But the other message here is that's a fairly long timeline from another perspective. If you think about the investment that went into both the drug and the diagnostic to gain this approval. The two package inserts reference each other. So in the Zelboraf drug label, it says that only patients who are detected with an FDA-approved test are recommended for treatment with this drug. And similarly, the drug is referenced in the package insert of the diagnostic. I will say that within 24 hours of this approval, there were a number of companies with laboratory-developed tests advertising that they had a test for Zelboraf patient selection, clearly not what the FDA intends with the label here, and nor were those tests clinically validated in the trials the way that we did. Just to give you more granularity about that, you're welcome to look at the package insert if you like that sort of detail, but over 25 assay performance studies with 600 unique specimens to verify the analytical performance. Moreover, the clinical performance, and that's most important, over 2,000 metastatic melanoma patients were tested, and those who were positive were shown to benefit from this drug. A highly reproducible test across multiple labs, and importantly, the FDA regulates the entire system, the hardware, the software, the reagents, and they do so on an ongoing annual reporting basis so that you have a standardized test that works really well. A little conundrum we had, because technology is moving so fast, the FDA requires that the gold standard be used as a comparator for this. The gold standard for DNA variation is Sanger sequencing, and it's actually a bronze standard these days. It's really not as good as what you can do with PCR, and we had to use the next generation of sequencing to show that Sanger was wrong and that our test was correct. But the other reason I bring this up, there are laboratories who use that technology as a diagnostic today. And clearly, that would be a problem for patients, that they could potentially be misdiagnosed. This is not yet routine by any means. There are many challenges, but the FDA is certainly working with us to try to make it more facile, both from the therapeutic and the drug and diagnostic development perspective.